The next presentation we have is QurCan Therapeutics. QurCan Therapeutics enables safe, repeatable IV delivery of RNA therapies to the brain via its non-viral TERP platform, solving a long-standing challenge in genetic medicine, crossing the blood-brain barrier without viral vectors. To present, we have Mohammed. Mohammed, over to you.
Thanks very much, Mobeen. And hello, everyone. Glad to present QurCan Therapeutics. Just briefly about the company itself. It's a University of Toronto spin-off biotech company, and we're very much focused on developing advanced genetic medicine for treatment of neurodegenerative diseases using our CNS-penetrating nanoparticle technology. And we're addressing one of the major barriers in CNS genetic medicine, I'm sure you're probably familiar with, is the blood-brain barrier. Is a physiological barrier in our brain that blocks out more than 99% of the injected doses having access to the brain. Today, in order to get the nucleotides to the brain, there's essentially two major routes. One is using AAVs or viral vectors, and because of the lingering toxicity, as you might know, it makes it more like the one and done scenario in the clinic. The other route is by injecting the nucleotides directly into the spinal cord, known as intrathecal dosing, and that path has its own clinical challenges, particularly because of its limited volume and frequency of dosing and poor brain penetration profile. So we were thinking how we can really create something that can be a breakthrough in genetic medicine, particularly for CNS neurodegenerative diseases, and impact a broader population of patients. And that, I think, the answer is to having some alternative avenue that allows us to repeat dose and also deliver any types of nucleotides so we can tackle different types of indications. And that's exactly what we've been, as I said, we've been working around. We've been successful in building and validating really the next generation nanoparticles. We call it TERP technology, or Target Engineered Responsive Polymer. It is essentially a hybrid polymer lipid nanoparticle system, very much differentiated with what we know of the LNP used in COVID vaccine, that allows non-immunogenic CNS delivery of nucleotides. And because it's non-immunogenic, you can do repeat dosing. So you can titrate the dose to get to the maximum therapeutic outcome. And on top of that, we can deliver any types of nucleotides, from small oligonucleotides to large mRNA and DNAs. So it creates a platform that you can plug and play and use this technology for a wide range of indications and clinical applications. And because it is a synthetic chemistry approach, without having any complex biologics involved, it's very easy to manufacture and scale. As I probably touched on, this technology has been very broadly and extensively validated across different species, from rodents to non-human primates. As you can see, this is a very cool data. Probably I must say this is the first of its kind data sets showing the large mRNA penetrating inside the brain after IV dosing. And that was done using our CTERP technology. You can see the signal of the mRNA under the machine. You can see that this is the signal of the mRNA in the brain. You can exactly see that a few hours post IV dosing, the mRNAs penetrate inside the brain. And more interestingly, unlike what you could potentially see with any other vehicles, you don't see that much accumulation into the liver tissue. It facilitates extrahepatic delivery and more exposure to CNS and brain area. One of the indications that we are very much interested in, and we believe that there is huge value to tackle, and CTERP can create a lot of value for it, is Dravet syndrome. It's a rare disease caused by loss of function mutation in a gene called SCN1A. Unfortunately, these patients are at the high risk of mortality, early mortality, and there's no cure or rescue. There are drugs available. All the drugs out there are symptomatic therapeutics. And as I said, no genetic medicine available. There's one in the clinic at phase 2 developed by Stoke Therapeutics. It's an antisense oligonucleotide. You can see the data here is an intrathecal formulation, so it can be injected in preclinical setting directly into the brain, or in the clinic through the IT dosing. And they showed when you inject it directly into mice brain, you can get about 85 to 90% survival. We package the same nucleotide in our CTERP technology and inject it systemically IV. At half the dose, we observe similar, survival efficacy. It really shows, and this data plus so many other ones, that the CTERP technology, because it's really give us the access to broader regions of the brain, we can penetrate in larger population of cells and number of cells and produce higher amounts of the target protein and have a better efficacy outcome at lower doses. And again, you can titrate, you can do repeat dosing, unlike what you could see with intrathecal or AAV. So we've been, over the past years, been able to significantly validate the platform and advance our internal TERP-enabled nucleotide therapeutics for rare indications. Okay, perfect. So yeah, we've, just repeating myself again, raised $70 million so far through the seed plus pharma partnership revenue, and advanced the pipeline to in vitro, in vivo POC, and currently asking for $30 million in financing to not only be able to identify 3 drug candidates for Dravet, spinocerebellar ataxia, and Rett syndrome, but also advance the lead program towards clinical POC. That would allow us to identify 3 drug candidates and hit multiple value inflection points. As you can see, we can not only be able to potentially.
Can you share your slides, please?
Oh. It sounds like my share icon is got offline, so I don't know if the setting has changed somehow.
I'll see what I can do here.
Yeah.
Okay, you can continue, I guess.
Yeah, for sure. Yeah, absolutely. So with the 30 million, as I said, we would be able to hit multiple value inflection points and not only be able to generate revenue through licensing of the asset or the platform, but we also could create exit scenarios through an acquisition after getting the access to the clinical POC data, like what we have seen for some of the M&A deals over the past few years.
You should be able to share your slides now, Mo.
Oh, there you go. Perfect.
Sorry about that.
No problem. Okay. We talked about this, and the team, last slide, is we've been really lucky, I would say, to be able to have amazing world leaders that are experts in the field of biotech and life science, people who've done this before built unicorn biotech companies, and highly committed to build another success stories, build products, advanced genetic medicines for patients, and send them home disease-free. With that being said, thanks very much for your time. My email is down here. Please do reach out to me if you're interested to learn more about our tech and the company. And if you have any questions, happy to answer.
Thanks, Mo. Thank you very much. Just maybe kicking it off with this question, and then we can wait for the chat if others have questions as well. Have you evaluated the safety profile of this technology?
Absolutely, a very good question. I guess safety is one of the major, as I said, bottlenecks in genetic medicine. Creating efficacy without causing immunogenicity. We have broadly explored the safety profile of the TERP nanoparticle in mouse, rats, rabbits, and dogs, non-human primates, under non-GLP and GLP setting. For instance, in GLP setting, the animals were dosed once weekly for 4 consecutive weeks, up to a month of dosing, and we observed no changes in liver enzyme panel, renal panel, blood chemistry. We were limited by the volume of injection. It's really called maximum feasible dose, without having any maximum tolerated dose. So it's one of the value that we bring in with the CTERP, that you can easily titrate without having any safety concerns. The immunogenicity also explored by third parties or US pharma partners and NIH facility, exposing the TERP in humans, blood sample donors, and exploring the immune cytokine excitements, and they observe no adverse immunogenic reaction or complement activation.
A question in the chat is, how are you allocating the funds?
So I'm going back to, so the fund, about, as I put it down here, like 7 million will geared towards getting at least 2 drug candidates nominated, going through the POC, and biodistribution studies in non-human primates. And I think that's the next value inflection point for the company. The reminder, which is close to 15 to 20 million, will be towards the IND enabling and the clinical POC for the lead program.
Great. And then one more question I have here is, what mechanisms do the nanoparticles use to cross the BBB?
Yeah, absolutely. This is a very novel mechanism. We call it dual targeted approach. So unlike the conventional methods that people were going after in getting engaged with a single receptors on the BBB endothelial cells, we are getting engaged with two different receptors on the endothelial cells. We call it dual targeted approach, that increase our chance of being detected by the receptors by more than twice as much. We've learned through the years that these two receptors that we've identified work in a synergistic manner and increase our chance of getting across the blood-brain barrier by folds as opposed to a single transcytosis mechanism.
Great, Mohammed. Thank you so much for your time and presenting. If you want to get in contact with QurCan, Mohammed, if you can just quickly share the contact us screen, and you can quickly get his email there. Thank you again for your time and presenting.
For sure. Thanks very much for the opportunity. Have a good day.